Hi, my name is Tristan Rainbow, and I worked on the Human Cloning Group 4 project. I helped research, and I really enjoyed this topic because I learned a lot about human cloning and science.
Hi, I'm Denise Bajami, and for the Group 4 project, I contributed parts of research, communication, and time management between the group and I for film management and a layout for our video.
My name is Esther Shin, and I contributed to this project by finding all of the research and typing out the bibliography and the ideas that we have talked about in this project. I was interested in this topic because the idea of human cloning was fairly unreal, but it was possible at the same time. And it was something that I never thoroughly researched in, so that was why I picked this topic in the beginning. Human genetics is defined as the study of the inheritance of characteristics by children from parents. Human genetics presents original and timely articles on all aspects of human genetics. It includes gene structure and organization. It links to human cloning, which is described as a number of different processes that can be used to produce genetically identical copies of a biological entity. The copy material, which has the same genetic makeup as the original, is referred to as a clone. The procedure for cloning is fairly complicated. It obtains organisms with the same genetic information. How's it done? You need to collect an egg cell from a donor, like a female sheep, mouse, or cat. Then you need to carefully remove the nucleus from the cell and collect another cell from the skin, or other tissue from another male or female donor of the same species. This is the animal that will be cloned. From the cell, you also need to remove the cell nucleus and place it into the empty egg. The egg obtained in such a way needs to be treated with a gentle electric shock. The egg should begin to divide and grow into a multicellular embryo. At this stage, the embryo needs to be implanted into the uterus of a surrogate mother. If the pregnancy develops normally and the animal is born, we have a clone.
History of human cloning. Researchers took a cell from an adult monkey and fused it with an inoculated egg cell. The embryo was allowed to develop for a time. Then its cells were grown in a culture dish. These cells, because they can differentiate to form any cell type, are called embryonic stem cells. This experiment showed that nuclear transfer in a primate, which researchers had tried for years without success, was possible. Opened the door to the possibility of human therapeutic cloning, creating individual-specific stem cells that could be used to treat or study diseases. In 2004, a group led by Woo Suk Hwang of Seoul Nation University in South Korea published a paper in the journal Science in which it claimed to have created a cloned human embryo in a test tube. However, an independent scientific committee later found no proof to support the claim, and in January 2006, Science announced that Hwang's paper had been retracted.
Human cloning safe? Scientists have found potentially definitive evidence that cloning is far too unsafe to be used in human reproduction. Its extreme high rate of death and the risk of development abnormalities from cloning makes cloning people unethical. Hidden genetic defects were found in otherwise healthy cloned animals in a study that could fatally undermine the arguments for losing control on the most controversial area of reproductive technology. David Humphrey, a member of the research team, said that by tagging certain genes, the scientists found that cloning appeared to upset a phenomenon known as genetic imprinting, where the genes on the chromosomes from one of the parents are switched on or off. Is cloning possible on humans? From a technical perspective, cloning humans and other primates is more difficult than in other mammals. One reason is that two proteins essential to cell division, known as spindle proteins, are located very close to the chromosomes in primate eggs. Scientists first used somatic cell nuclear transfer to create a human embryo that could be used as a source of embryonic stem cells. The resulting stem cell lines were specific to the patient they came from. Researchers took a skin cell from the patient and fused it with a donated egg cell. Key to the success of the experiment were modifications to the culture liquid in which the procedure was done and to the series of the electrical pulses used to stimulate the egg to begin dividing. Despite several highly publicized claims, human cloning still appears to be fiction. There is currently no solid scientific evidence that anyone has cloned human embryoes.
As we introduced our research question of human cloning, we came up with the introduction of human genetics. And from here, we connected that to that idea of human cloning, and from there, we came up with all of these subtopics that we used in our research. First, we came up with procedures to gain a basic understanding, then history to see if anyone even tried using this, and if it was still being used today, which connected to an unethical, and if it was ethical. Then we came up with, is this even safe? Is this even legal? Can this hurt? And we came up, and as we researched, we saw that it had a very high rate of death. Then we came up with another subtopic, and we asked, is this even possible on humans? And then it said that it barely worked on animals. So it could be safe to assume that human cloning could be possible, but not anytime soon.